Hey, everyone. Welcome, welcome. All right, it's great to see you guys. Feels like the weeks go really quickly, doesn't it? Yep. Here we are, Tuesday again already. All right. Hey, anybody have any questions? All right. Doing a great job. Let me get us started here. All right, let me know if you cannot see that where it says biology. Okay, hold on one second. I gotta do one more quick thing here. Sorry. Okay. All right. So I just want to give you a heads up. In our live class in 2 weeks, we're going to be doing an activity together in class. Now, you don't have to do the activity. It does need some supplies that you have to order. So if you can't get this stuff or you choose not to, that's fine. You can observe. I just think it's more fun if you do it on your own, if we actually do the lab. So you'll see this supplies list. In the major supply list and also at the beginning of the chapters, there's supplies for each week, but I just wanted to show it to you now. So the things that you will need are safety goggles, this chemical, bromothymol blue, which we will use another time later in the year. So you won't be buying it just for one lab. Here are some places that you can buy it online. You might also be able to get it if you have a science supply store near you. Some people do, some don't. You need 3 small jars or test tubes with caps. So the small jars have to have a lid. So does the test tubes. Either are fine. You can use like a little baby food jar or a little mason jar, something like that. Even a jar that was left from, you could use like a water bottle. That would work. Something with a lid on it. Small is better. So you need a test tube rack if you're using test tubes. You'll need a drinking straw and a plant that grows underwater. So you can get those at aquarium stores. In fact, if you go into an aquarium store and say, I just need a really tiny piece of a water plant for a science experiment, they might just give it to you. I've had students tell me before that you don't need the whole plant. You just need a chunk of it, like a little branch of it. I use one called hornwort. You might also be able to get an underwater plant if you have a water source near your home, but it has to be one that grows completely underwater. And then you need some water. Okay. So don't wait till the very last minute. If you have to order the bromothymol blue, it may take a little bit of time to get there. Amazon is the most expensive place, but it also is somewhere you can get for, right immediately, right away. All right. Any questions about that? Okay. Let's talk about this lab that you guys have been working on. So what we did here is we had different bags with a paper towel inside. We put some seeds in the bag. We put some peroxide of different concentrations in that bag, sealed it up, and watched what happened over time. Now, remember what a controlled experiment is. Was this one? Was this a controlled experiment? Bethany
Yes,
said yes. Yep. You say yes, Luke. Why? What made
because
it?
we put some factors in our experiment were different than others. For example, in some baggies with some seeds we put in some bags there were carbon dioxide in others they weren't or less.
Okay, so
Sorry, I mean
yeah, I
um
understand. I think I understand what you're
sorry.
saying. What makes something controlled is when you only change one thing. There's only one variable. So now knowing that definition, do you think this was a controlled experiment?
Yes, because there was a bag that had no carbon
Yes.
dioxide, sorry.
Peroxide is what you're thinking. Hydrogen peroxide.
Peroxide.
Yeah, yeah,
So
yeah. We
many
had a
terms.
control group. Yeah, you're absolutely right. So that changes everything, Luke. I was thinking carbon dioxide, but it was hydrogen peroxide. Same thing. But yeah, your answer is correct. That's all we changed. All we changed was the concentration of hydrogen peroxide. Now, the truth is there were some variables that were changed. It's really hard to do a completely controlled experiment, but we did the best that we could. And of course, as a group, as a class, it wasn't all that controlled because we had many different variables from one home to another. So Maria's house might have been one temperature where Zion's house was another. There might have been a humidity level in Luke's that was different than Bethany's, right? So that makes a big difference. So as a group, it wasn't controlled, but individually it was. So let's look at this experiment and let's talk through the writing of a lab report as we would do that if we were doing it in this one. So here are the first things that are on a lab sheet that you can use to help you to write a lab report. So we start out with the question. Usually we observe something and then we ask a question about it. So our question this time would be something like this. Which concentration of peroxide will cause the seeds to germinate fastest? And then we develop an experiment to try to test that. We want to make a prediction about what we think is going to happen. Which concentration do we think is going to cause the seeds to germinate fastest? Now, the prediction is pretty informal. You don't always have a prediction in a lab report, but when you write your hypothesis in if-then format, you do. So I want to look at the possible hypotheses and and have our hypothesis include what we're going to do and then what we predict is going to happen if-then format. So here are a couple hypotheses from students I've had in previous years. So I want to just evaluate these and say what's good about them and what's not so good about them. Let's look at the first one. If seeds are moistened with the highest level of hydrogen peroxide, then the seeds will germinate faster. All right, so that's pretty good. What could I do to make that better? One thing I really like about all these is that they are if-then format. If they're moistened with the highest level, then they will germinate faster. If the 5 milliliter bag works better for pepper seeds, then it is the best growing solution. If there are higher concentrations, then the seeds will grow faster. Now, they're, like I said, they are good because they're in if-then format, but we could improve on these. Now, remember, when you have the if-then format, the if is your independent variable. The then is the dependent variable. The independent variable is the thing you change. The dependent is the one you measure. Basically, we're looking at this in an if I do something, then something will happen kind of way. So remember this guy, our friend.
Independent variables are the ones you change.
That's really all we need to remember. Just that part of the song. Independent variables are the ones you change. If that's all you remember, you know the rest, right? Because you know what controls are. Controls, you control them. You keep them the same. So taking a look at this little experiment here, how light affects plant growth. So what is the independent variable? Independent variable is the one you change. What are we changing in this experiment?
The amount of sunlight.
The amount of sunlight?
Yeah, excellent. The amount of sunlight is our independent variable. And then our dependent variable is going to be the growth of the plant. So let's write a hypothesis using those independent and dependent variables, the if and the then. If something is done, then something will happen. Can somebody suggest an if part of this?
If we use a lot more sunlight than usual, it will grow faster.
Okay, excellent, excellent. If the plant is given more sunlight, then the plant will grow faster, taller. Whatever we use in our then has to be measurable. So is taller measurable? Yeah, we can use a ruler. Is faster measurable? Yes, we can track it by time. So we just want to make sure that we have our if tells us what we're going to do, and our then is a measurable thing that we can actually tell is happening. All right, now I can also do this the opposite way. I could write a hypothesis that says, if the plant is deprived of sunlight, then the plant will die. I've got my independent variable depriving the plant of sunlight, and my dependent variable, the plant dying. It's something that I can measure. So I can do that both ways. All right, so let's go back to our hypotheses for our seeds again. So let's incorporate the independent variable into the if. We're not going to do this second part. We'll just do the if I do something. So let's use this first one. If seeds are moistened with the highest level of hydrogen peroxide. That's okay. There's something I could do to make that better. What can I do to make that better? more specific? Bethany?
Is my mic working?
Yep.
Okay, good. Maybe the type of seeds you're using?
Yep, we could put the type of seeds in there. If bean seeds are moistened, what else? That's a good one.
Where you put the seeds, like the amount of sunlight.
The amount of sunlight, possibly, that's a good point. So we're not really, sunlight isn't really our independent variable though. Remember, just what we want in the if is the dependent or the independent variable. The sunlight is going to be something that's going to be a controlled part of our experiment. We have to think about it. That's important. What about instead of saying highest level of peroxide, we told what that level was, right? That would give us, that'd be more specific. So if the seeds are moistened, and let's say if we put bean seeds in there, if bean seeds are moistened with a solution made with 30 milliliters hydrogen peroxide and 250 milliliters of water. Okay, that's pretty good right there. All right, now let's look at this one here. If the 5 milliliter bag works better for pepper seeds, there's really a lot wrong with that one. One thing that's wrong with that one is the bag working better isn't really something we're doing. It isn't something, it isn't the independent variable. How could the bag be working better? Right? So we could do something like this. If seeds are placed into the bag with 5 milliliters of hydrogen peroxide and 250 milliliters of water. But that's still not great. If you read that, what would you think is going to happen? What would you think the experiment was going to be?
Probably something to do with the bag.
Yeah, probably something to do with the bag. Probably the bag, it sounds to me like the bag is going to be full of water. All right, like I'm going to have a bag full of 250 milliliters of water, which is a lot of water, and then 5 milliliters peroxide. I'm going to dump those seeds into it. So I don't want to just say if they're placed into the bag with all that water. So I can make it better by saying if they're placed into a bag and moistened with a solution made with that water. That's a possibility. And then the third one, if there are higher concentrations of peroxide present, again, that's not really something, that's not really specific enough. It's not really something we do. So again, this top one right here, if seeds are moistened with a solution made with X milliliters of peroxide and 250 milliliters of water, that's probably the better way to write that. All right, now let's do the then. This is the dependent variable. So using the first one, if seeds are moistened with a solution made with 30 milliliters of hydrogen peroxide and 250 milliliters of water, then the seeds will germinate faster. So our second part, our then part, our dependent variable has to be something we can measure. Is this measurable? It is, but it's not really clear what we're comparing it to. Faster than what? Seeds will germinate faster than seeds that are kept in the dark, faster than seeds that are, thrown in the trash, faster than what? So we want to have something more specific than that. We want to say then the seeds will germinate faster than the other tested solutions. Write that. All right, the second one then. If seeds are placed into a bag and moistened with a solution of 5 milliliters, then it is the best growing solution for seed germination. That's not really measurable. Best growing solution. What are we measuring there? We don't really have anything we're measuring. So we don't want to use words like best and better and those kind of things. And then the third one, then the seeds will grow faster. All right, we're not really testing growth. That's not really what we're looking for, is growing faster. We're testing germination. So we don't want to say the seeds will grow faster. We want to say something about the germination. All right, does that make sense for how we write a hypothesis? All right, another thing to keep in mind, when you're writing a lab report, you want to use passive voice. So what does that mean? That means instead of saying I did something, you say something was done. Instead of saying I put the seeds in the bag, you say the seeds were placed in the bag. Instead of saying I added peroxide, you say peroxide was added. Here, instead of saying Paul used this method, you say the method was tested. So Most teachers in most subjects are going to want you to use active voice. Lab report is different. Lab reports use that the passive voice. And the reason for that is the person doing the lab, doing the work, that's not the important part of the lab. The important part is the process. It's what you're learning, what's being done. So instead of saying, if I give the plant more sunlight, then the plant will grow taller, we want to say, if the plant is given more sunlight. You see that? This is active. I'm doing it. This is passive. If I deprive the plant of sunlight, we want to say, if the plant is deprived of sunlight, then the plant will die. All right? Passive voice. Now, please remember, this is not the only way that you can write a hypothesis. You don't have to use if then, but it's almost always best to do so if you can, because it tells you what you're going to do and it tells you what you're going to look for. Some resources suggest that you even use a 3-part hypothesis, where you say, if something is done, then something will happen because of this or due to this. Often this is called the if then because format. For my class, I just want you to write if then, and I'm going to strongly recommend that you do that. Whatever you write it, a hypothesis always includes what you expect to happen. It's clear and understandable, testable and measurable, and it contains both the independent and the dependent variable. All right, now that we talked about hypothesis, let's go back to our experiment that we're doing. And let's say that this is the hypothesis that we decided we're going to do. If seeds are moistened with a solution made with 300 or 30 milliliters of hydrogen peroxide and 250 milliliters of water, then the seeds will germinate faster than other tested concentrations. All right. Then after we put our hypothesis in, the next 2 things are our supplies and our procedure. It's really important that you put every single thing that you used. Everything. If you forget to write paper towels in your supplies list, can someone reproduce it exactly the way you did? No, they can't. It's like leaving sugar out of a cake recipe. It's not going to turn out, right? You want to make sure you have every single thing that you use. The same thing with the procedure. You have to list every single thing that you did. If you forget to say, close the top of the Ziploc bag, then the person who comes along after you is not going to close the top of the Ziploc bag, and it's going to dry out and it won't work. So you need to tell them what to do, exactly what they need to do in order to reproduce your experiment completely. And the best way to write your procedure is like a recipe. Do this, then do this, then do this. You want to number your steps. Don't repeat your materials list. And please don't just copy and paste what I tell you you can use. So like in this one, if I said you can use bean seeds or pea seeds or radish seeds or broccoli seeds, don't put that in your materials list. Don't say you can use bean seeds or, don't copy and paste. You have to say what you did. These are the things that you used. And this, I think, is really important. When you're done, have someone proofread your lab report. Do they understand it? Do they know what they can do? Could they reproduce it without you even being in the room? Right? Without you being present. That's a good lab report. If someone can completely reproduce your experiment the way that you did it. All right. Then we get to results. The next thing after the procedure is what the results were. Results is another way to write what you observed as the experiment was going on. So you guys did this. You gave me your observations, and there were a wide range of answers that we had here. Now, some of them had a lot of detail. Some of them were only a sentence or 2. Not very many of you gave me a chart. Charts are really great. Charts are often what you want. In fact, if you can use a chart, do. If you can use a graph, do. Because they're a good pictorial, quick way for your reader to be able to figure out what it is that you're trying to say. All right, so I took pictures. I did this lab, and I took pictures of my results. So I'm going to use them. We're going to fill out this chart. We're going to evaluate the results as we go. All right, so here they are. Now, I put them in the bags on day 1. I didn't see anything happen on day 2. So these are my results from day 3. So somebody evaluate these results.
It seems that the seeds are slightly opening up.
Which ones? Which ones do you think germinated first?
Low.
Yeah, yeah, almost all of them. In fact, all of them. This one maybe not, but all the rest of them have germination. We've got a couple little tiny ones here, but the low definitely look the best, had the most germinate. And then the control had no germination at all. These look just the same as they did the day I put them in. All right, so here's how I filled out my chart. All right, I saw, no, this should say germination. It should not say growth. I'm sorry about that. I forgot to change that. So no germination, no germination, no germination of the control on any of the 3 days. In our peroxide, there was germination and a little bit of growth on day 3. All the rest of them had no growth, no germination, no germination on days 1 and 2. There was a little bit of germination on the medium and the high, but there was more on the low peroxide day. All right, so now this was my day 4. Now here's the thing. What we were testing here was germination. So really, once we got to day 3 here and we had germination, this experiment could have ended. It's over. We figured out which germinate so that we don't need to go any farther. I decided to continue the lab for a few more days just so we'd have a little bit more data. So you can see here on day 4, we've got more growth. There's definitely more in the low bag than there were on any of the other days. Now, that's how I charted that. I measured it. Remember, we want to measure wherever we can. So we're about 2.5 centimeters of growth in the low peroxide. The medium had about 0.5 centimeter and the high had about 1.5 centimeters. Then the next day, this is day 5, the low was definitely better. We got some branching of those roots there. I put that down here. More growth and root branching. Small amount of growth, small amount of growth, small amount of growth. And then finally, my last day, day 6 and 7 were about the same. They showed the same pattern. Look at all the growth that we have here. So I've completed my chart. This is my data. This is all you have to turn in for data. You don't talk about it. You don't evaluate it. You just simply say, this is what happened in the experiment. So we could do it this way. Instead of a chart, we could do this. The seeds in the control. This is all of your results. This is all the results of the whole, both sections of biology as of Friday of last week. So the seeds in the control bag germinated first for 4 of you. The seeds in the low concentration germinated first for 6 of you. In the medium for 3, in the high for 1, and none germinated for 1 of you. All right, now that's a paragraph. That's okay. But look when I take that same data and put it in a table. How much easier it is to figure out which germinated first. For 6 of you, the low concentration germinated first. Much easier to see. And if I put it in a graph like this, now it's really easy. So you can see how the table and the graph really help. And again, this is all you need for your results. You don't need anything else. The next step after results is where you talk about things. This is the discussion section. So we would talk about them. We would say, do we have enough data to come to a conclusion? Was your hypothesis supported by your data? What more do you think you want to do? What do you think might have gone wrong? What might you change in the future? What other factors could have been at play? You want to really think through and be very, very specific in your discussion section about what could have happened, what else you could test, and what you learned as a result of that. All right, so conclusion. Let's talk about our conclusion. Remember, this was our hypothesis. Then the seeds will germinate faster than other tested concentrations. So if this was our hypothesis and the solutions we used were 5, 15, and 30, using the results that we got, we could say, since the lowest concentration gave the fastest germination, higher concentrations of peroxide do not appear to improve germination in seeds. It did not prove our hypothesis correct. So that's our conclusion. That's really all you need in a conclusion, is just simply tell what happened. The bulk of all the talking through happens in the discussion section. Now, we could also have written this a little bit differently. We could have said, since the highest concentration gave the slowest germination, higher concentrations don't improve. Or since the lowest gave the fastest germination, low concentrations do appear to improve germination in seeds. So there's a lot of ways you can write that. You just need to make sure that they are talking about comparing it to your own hypothesis. All right. Now, this shows the results for the last couple of years of my biology classes. So you can kind of see here how now it's beneficial to have a bigger sample size. So what we can conclude looking over the past 3 years, we can see that the low is better. We might conclude based on that, that the low concentration provides of peroxide improves germination in seeds. All right. And with that, we just wrote a lab report. So you guys did the lab report. You just did it in steps. You did it in little stages in one lesson at a time. But that's basically what a lab report is all those pieces put together in one. You'll be doing a lab report in a few weeks. You'll follow the same exact procedure. I'll talk a little bit about more about it before I assign it, but this is what you do. Just remember, when you write a lab report, it has to have a title. The title tells about what the experiment is about. You don't want to be cute. You don't want to be funny. This is a technical document. So you want to have a title that tells what the experiment is about, a purpose that tells why you did it. You want to have the hypothesis, which we just talked a lot about. You want to have your materials list and your procedure so that somebody else could completely reproduce it. You want to have your results section and only results. You want to have your discussion and you want to have your conclusion. All right. Anybody have any questions about our experiment that we did or writing a lab report? All right. We're going to spend the rest of our time doing a quick review about the content that you guys had in your lessons last week with a little bit more. So when you hear the term population, I'm sorry. I want to talk about this first. This is one of the quick lab activities that was an option for you. I don't know if any of you guys did that population density project where you put a lot of seeds into a cup and then you allowed them to germinate. If anybody did those, I did, and I took some pictures. So I want to show you. So days 1 and 2, I really didn't see anything. It was just soil. Day 3, I started seeing a little bit. This is what I saw on day 4. So all these seeds are coming up. This was the next day, day 5. And this was day 7, 2 days later. All right. Now, how well do you think these are going to continue to grow? Do you think they'll keep getting bigger and bigger and bigger like that at that rate?
No, because of the size of the pot.
Yeah. Yep. The size of the pot's going to slow the growth down. There's not enough soil to be giving them the nutrients and the water that they need. And there's simply not enough room. They're going to be crowded out. The roots are going to get too crowded and it won't be able to grow. So this is kind of a fun, I'd recommend you do these labs if you can, even the ones that are not required, because I think they're kind of fun to do. Now, one thing that's wrong with this experiment right here is I didn't have a control group. So if I had a control group that only had one seed in it, I might see that one plant grew very differently than the ones that are crowded in this pot, in this cup. So that is something to consider. All right, I got a couple population jokes for you. If the human population held hands around the equator, a significant portion of them would drown. And I like this one. Which country has the fastest growing population? The country is Ireland, because every day it's Dublin. All right. What do you think of when you hear this term, population growth?
The rise of humans,
Rise
population.
Rise of human population. I probably biased you a little bit with the picture there, which is all humans. Population growth can apply to any living thing. The population of bacteria as it grows, we would call population growth. So do any of you guys think that the world is becoming overpopulated? That we're getting too many people on the planet?
No.
No, you probably heard enough that we're not. Our population growth has really slowed. Well, about 20 years ago, what everybody was saying was that, oh, we can't possibly support any more people. We're reaching the capacity of the earth to support humans. Well, that hasn't actually happened at all. We'll talk about that in just a minute. So let's take a look at the population of the world. This is the population from 1790 until a little after 2010. So what trend do you see in that time? Bethany?
Very large increase in
Yeah.
population.
Yeah, we have what's called exponential growth, where it no. So this is the U.S. population, not the world. So this goes up like this. This is a J-shaped curve. You have to kind of use your imagination to picture a J, but that's what they call it, a J-shaped curve. That means there's nothing slowing growth down. We're continuing to grow and grow and grow and grow with nothing slowing it down. Now, if we compare that to the world population, you can see that the population of the world is increasing in most places. In North America, it's gone up slightly, but the total number of people is really only part of the equation. The other thing that we need to look at is how rapidly the population is growing. So what do you see here? This is US population growth. What's happening to growth in the US?
It seems like it's decreasing.
Yeah. So how can the population be going up, but the growth is going down?
If I had to make a guess, I would say maybe the food that we have here, like the fast food probably has impact on the youth, has impact on people. Our meat, kind of the farm raised, we don't really have really good our farm raised meat isn't that good. You're feeding chickens, you're feeding chickens you're feeding chickens things that aren't on their diet, for example.
Right?
So that could be a reason why the population could be decreasing, because food could cause cancer.
Okay, those are all really good points, Luke. But remember, the population is not decreasing. So let's look back here again. The population is going up. It's population growth that is decreasing. Everything you said is true and probably is playing a role here. But I'm wondering, how can population growth be dropping while the population itself is increasing?
What's the difference between the two of them?
That's a good question. What do you think?
No clue.
That's why you asked, right? So population is how many people there are in the US, if that's what we're talking about here. How many people are here? Population growth is how fast is that number increasing? Well, what's happening now is that number is not increasing as fast. We're actually losing more people. We're not replacing people at the same rate that we used to replace them when they die. So the speed at which it's increasing, the rate at which it's increasing is going down. Bethany?
Maybe people are taking more time to have children, and that's why.
So we'll talk about the whys of that in just a second. That's a definite possibility here. Yeah. So this one goes up to 2019. So if we look at the world, that's the US. If we look at the world, this is the population growth rate. Now, in order to be stable, the population growth rate has to be every 2 people on the planet have to have 2 children, right? That keeps things flat. If every 2 people are having 10 children, then we're going to have a big population growth. The growth rate is going to go up. If every 2 people on the planet have 1 child, the growth rate is going to go down. We're not replacing people as they die. Does that make sense? So what the population growth rate really needs to be stable is 2.2. It needs to be a little more than 2 people have 2 children. It needs to be a little more than that. But you can see in this graph, that's not happening. The growth rate since 1960 has trended down. So this is about 1960 right here. Get my red pointer right here. It has trended down. And this is about where we are right now. This is the UN prediction of what is going to happen. They predict that by just maybe 2090, there will be 0 population growth. That means we will not be increasing the number of people on our planet anymore. We will level out. The number of people on the planet will stay the same. We'll exactly replace the number of people who die. So we are on the way in that direction right now. So think of it this way. Right now, the growth rate is about 1.1%, right? 1.1% of the population we have today is not the same as 1.1% of the population last year. So think about if you get an allowance from your parents and they said you can have 1.1% of $100 or 1.1% of $1,000, which would you have? Or how about this? If they said you can have 1% of mom and dad's salary this year or 1.1% of mom and dad's salary next year. Well, mom and dad are probably going to get a raise at the end of the year. So their salary is probably going to be a little higher. You'd be better off taking it of that. So you can see how it happens. As the population continues to grow, the growth rate has continued to slow down. We're just not replacing people as fast as we used to. I have a little thing here, the same kind of chart, but it shows in a time lapse, how that happened. So here we are, 1960s, 70s, 80s. Now you can see things really starting to drop until we get to about where we are today. And now they're predicting that the population growth is going to level out before 2100 and drop below zero. So we will not be replacing people the way that we did before. So that's kind of interesting. Now, just for fun, let's look what happened here. So right about here, this is about the year zero. This is about the year, 1000. Right about here, right about in the mid 14th century, we had a little drop in people, but then it started taking this upward shoot. We now have a little over 8 billion people on the planet right now. All right, so they estimate that around 1800, around the year 1800, the population was about 1 billion people. So that tells us that on average, the population grew really, really slowly from the beginning, from about 10,000 BC until about 1700. Then after 1800, everything changed. The world population was 1 billion in 1800. It has increased eightfold since then. So they say that around 108 billion people have ever lived on our planet, total. That means that the population we have today makes up about 6% of all the people who were ever born. Now, why do you think population right here started increasing dramatically? Any ideas? There's a lot of factors here. Bethany.
Maybe like a improved lifespan.
Yep. Why? What happened to improve lifespan?
Well, like advances in medical technology, better food, and
Yep.
stuff like that.
Fewer people are dying from
Industrial Revolution?
diseases. Yep.
Was that around there, the Industrial Revolution?
Yep. Right around the Industrial Revolution. Yep. So life became, we moved from the farms where people did what was called subsistence living, which means they barely had enough to eat. And we moved into the farm, into the cities at the Industrial Revolution. That changed a lot. One major factor that changed all this right here is what we call child mortality rate. So this is a little bit hard to see. But if we take a look here at across the entire historical sample from about 400 BC is where this starts up until 2017. On average, right about at the beginning here, all this time here where it was really pretty flat and then it started to drop, right during this time, about 1 out of every 3 babies died before they were a year old. Now think about that, 1 out of every 3 before they were a year, and almost half before they became adults. Now, that means that just as recently as 1950, the global mortality rates in children were 5 times higher than they are today. So on the very right of the chart, this part right here, we can see that it drops. Right now, the global infant mortality rate is not quite 3%. So it went from 33% to 3% in not very much time. And 4 out of every 100 die before they reach adulthood, where it used to be half. Used to be 50%. Now it's 4%. Pretty significant. So that change, that's the major factor why we saw that big upward spike that we did. Now, one of the questions people ask is, just how many people can live on the earth. Nobody's really sure. They've made estimates throughout time. Those estimates have ranged anywhere from, oh, we can only hold 500 million to more than 1 trillion. So right now we have a little over 8 billion people on the planet. So obviously 500 million wasn't right. They were wrong with that. And if it's 1 trillion, if the orange is 1 trillion, the blue right there is what we have currently. So you can see we can still grow a lot if the trillion is correct. The thing is, we keep doing things that increase the number of people that the earth can hold. So some of the things Bethany mentioned, we keep doing things so that we keep people healthy longer with some of the medical advances that we've made. We have better farming practices, so we're producing more food than ever before. We're using fertilizer, which is getting bigger yields out of our crops. We're building up. So taller buildings mean there's more space for people. The question becomes, what do people really need in order to be able to survive? And that's a big question and really not an easy one to answer. So we look at things like population dynamics to help us to figure out the answers to questions like this. This is the field of study that looks at how populations change over time. And of course, this applies not just to human populations. It applies to all populations of all living organisms. So when we're talking about a population, what we mean by a population is a group of organisms belonging to the same species that live in the same area. I always remember a bowl of popcorn. I think population, popcorn, those start with pop. When you've got a bowl of popcorn, they're all the same. They're all popcorn in that bowl. They're all in the same place at the same time. They're in that bowl right now. A population is all the same species living in the same area at the same time. Now, what's a species? Anybody remember what a species is? A species is a group of, oh, go ahead, Bethany.
A species is a group of like similar organisms, all part of the same group of organisms living together. I
Yep.
think. Is that how
Yep.
you say that?
You're almost right, almost done. You're on the right track. What they have to do, it's a group of organisms that can breed. They're able to have fertile offspring, meaning they're able to have babies that can go on to have babies. So like, you know, a dog can have babies with another dog, and then those babies are able to have babies. But a dog can't have babies with a cat, right? We don't have any way to do that. So a dog and a cat are two different species. All right. So some of the characteristics of a population, that group that's the same species in the same place at the same time, some of the characteristics of the population are where they are, their geographic distribution, how many there are, which is their population density, and how fast or how slow the population is growing. So let's quickly look at all three of those. The first is geographic distribution. It's the area that the population lives in. For instance, you're not going to find polar bears in Costa Rica, and you're not going to find tree frogs in the Arctic. Where they are, that's part of their population dynamics. It's part of what makes the population grow or not. The density is how many there are. If we've got a large geographic distribution, but a small population, so big area, small population, everybody has a lot of space. You've probably got some farmland around you. Most of our states have at least a little bit of farmland. That's an example of a lot of space with a small population. You probably also got a city nearby where there's not a lot of space and a whole lot of people all packed into a pretty tight space. That's a small geographic distribution and a large population density. And then the third is the growth rate. This is what we were talking about before. This is the way the number of individuals in that population changes over time. So our population is changing more slowly over time because we're going down. This one in the picture there, the population is increasing slowly over time until something happens to slow that population down a little bit. Now, population size depends on some things. It depends on who's born. It depends on who dies. And it depends on the people or the organisms that move in or that move out. So those terms, moving in and moving out, we use the words immigration and emigration to define those terms. So means moving in. Immigration means you're moving into an area. It's the movement of individuals into an area, and that increases the population in that area. Emigration is moving out of an area. That decreases the population size. And they always go together. You have to move out of somewhere in order to move into somewhere else, right? You don't come from nothing and you don't go to nothing. You emigrate and then you immigrate into the same place. Now, if there is a group, if there's an area and there's no immigration and no emigration happening, then the population growth depends entirely on birth and death rates. When they're the same, size of the population stays the same. When death rate is higher, population goes down. When birth rate is higher, population goes up. Now, there's some factors that lead to immigration and emigration. Can you name some? What makes an organism want to leave one area and go somewhere else?
Maybe if there's not enough food or resources.
Yep, there you go. In a nutshell, resources is what it comes down to. Things like food, a better quality habitat, mates, water, fewer predators, whatever makes life bad in one area, they try to escape to go to an area where those kinds of things don't happen. All right, a couple other things I want to make sure that you understand are these kinds of growth. So we talked a little bit about exponential growth a minute ago, which is that J-shaped curve that goes up this way. So take a look at this growth of the rabbit population. Now, if I start out with 2 rabbits, I have a male rabbit and a female rabbit. Doesn't take very long before I have 100 rabbits, right? Rabbits breed quickly. But what if I don't have enough food for 100 rabbits? What if there's not enough shelter or there's not enough water? They're not going to grow as quickly. Something's going to slow their growth rate. Well, when there's plenty of everything for everyone, this kind of growth happens. This exponential growth happens. Now, this doesn't depend on how many there are in the population. Just as long as there's enough resources, there could be 10 rabbits or there could be 1,000, as long as there is enough for each of them. This is exponential growth. It just gets bigger and bigger and bigger population over time. It happens under ideal conditions with unlimited resources. And it doesn't depend on the total number of individuals in a population. This little cartoon says it all. We are the classic example of exponential growth. 1 + 1 equals 53,671,821,309, right? All right, so look at this chart right here. You can see that the number of rabbits just keeps doubling. This is exponential growth. Nothing is slowing down the growth of the rabbits. Now, some factors that lead to exponential growth, basically anything where there's plenty of resources. As the resources come back in an area, even if it's been destroyed, then the organisms will grow. And this is that J-shaped curve that exponential growth gives us. So the population tends to grow slowly at first, but then faster and faster and faster over time. We saw that in that human population growth where it was slow, 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 and then took that sharp spike. All right, now this will continue until something makes it stop. What stops exponential growth?
Maybe when it rises so much that there's not enough resources for all the people.
Yeah, something is gonna, yeah. There's a number of factors that could stop that exponential growth from happening. We call the things that stop exponential growth limiting resources. And there really only has to be one. We have this thing in population dynamics that's called the law of the minimum. Basically, that tells us growth isn't limited by all of the resources. It's limited by the one that's the scarcest. So think about this barrel here. How much water can you put in the barrel? Well, you can only fill this barrel up to here, up to the shortest board. This board right here is the limiting factor in this barrel. Well, if we then use the same barrel analogy and we think of the things that plants need in order to grow, what's the limiting factor? It's going to be whatever this one is under here. We can't see what it is, but there's something, maybe light, that it doesn't have enough of. No matter how much sulfur it gets, no matter how much magnesium, how much boron, how much nitrogen. Oh, it's not light because there's light right there. Well, no matter how much of all these other things, it doesn't matter. Even if the plant gets all of these other things, if it doesn't have enough of this right here, it's not going to do well. The limiting factor is the thing that is going to slow that growth rate. So when that happens, when there's some limiting factor that slows down the growth, that slows down that exponential growth, we say that it has reached its carrying capacity. Carrying capacity is the largest number of individuals the environment can support over time. So we talked about what some of those factors are that limit that. The last thing that I want to show you before we run out of time here is the difference between logistic growth and exponential. So exponential, that's where it just goes up and up and up and up. Nothing slows the growth in exponential, just keeps going up. That's a J-shaped curve. Once it hits carrying capacity, it flattens out. That is an S-shaped curve, and that's called logistic growth. So when logistic growth is graphed like that, it looks a little bit like an S. It begins a lot like the exponential. See, it begins like that, but then something happened. Something happened to decrease the growth rate so that it could, not continue on and on forever and ever. No population can continue growing forever and ever without ever reaching carrying capacity. Now, we haven't reached carrying capacity on our Earth yet. We can still take more, lots and lots more people. So we haven't reached carrying capacity. When we do, it's probably going to end up making a graph that looks like this. So let's say we've got a population of bacteria, and the bacteria are growing, and then they use up all their food. Well, then they're going to overshoot their carrying capacity, use up all their food, and then they're going to die off. Well, now there's plenty of food again, so they're going to grow. Well, now they've eaten too much, and then they're going to die. And now there's plenty of food again, so they're going to keep growing. It'll do that kind of thing. It'll overshoot and undershoot. And this is what the graph usually ends up looking like. It lets us see the average number. This line right here would be the average number that the environment can support. All right, let me see what I want to cover because we're just about out of time. Okay, so some of the things that limit population growth. There's really two main categories of limiting factors. There are things that are alive or once were alive, and things that are not and never were alive. Do you guys remember what those are called? What do we call something that is not and never was alive? What kind of factor? So they would be called biotic and abiotic factors. Things that never were alive are called abiotic. So like rocks and soil, those are abiotic. Biotic are things that are and once were alive. So when we look at these two main categories, we have density dependent. That means that depends on how many there are. And then density independent, that doesn't depend on who they are. So density dependent would be things like competitors, parasites, diseases, predation. Those are things that are dependent on the population size. Because if you think about it, a parasite is more likely to spread when there's a big crowd of a population than when the population is spread pretty far and there aren't very many of them. So the other one would be density independent factors. Let me find that. Density dependent. Oh, here they are. These would be density independent factors. These are ones, doesn't really matter how big the population is. It could be a population of 1, it could be a population of 1,000. If you've got a weather situation, a natural disaster, or human activity, it's not going to matter how big the population is. It's still going to have an effect on it. And we're going to have to stop there because we are out of time and I'm losing my voice. Anybody have any questions? All right, keep up the good work, everybody. All right, terrific, Chris. Thank you. All right, have a good week. I'll see you next time. Thanks for coming, everybody. Bye.